Welcome everyone. Today I will be going to talk about three experiments that were performed by Alan Hodgkin and Andrew Huxley back to back in 1952 that had brought them Nobel Prize in 1963 in physiology or medicine. Now at first I will explain the experiment 1. To understand this experiment, we have to understand these 2 points at first. The first point is voltage clamp method. This method was discovered in 1940 by Kenneth Cole and it was applied by Alan Hodgkin and Andrew Huxley in late 1940s. By this method, we can actually control or fix the membrane potential of a neuron. The voltage clamp means you can fix this membrane potential or voltage. Now, this point I have already discussed in my previous video. Now, also I want to mention here that resting potential means if you measure the membrane potential in an unexcited neuron, it is called resting potential. Generally, it is minus 70 millivolt in value. So minus 70 millivolt. Now when the membrane potential increases over the resting potential, we call it depolarization, and the membrane potential that is lower than resting potential, we call it hyperpolarization. This graph represents the x-axis in millisecond and the y-axis in millivolt. That means this is the membrane potential that is applied on the neurons. Now Alan Hodgkin and Andrew Huxley take the help of squeezed giant action because the diameter is higher or larger, so that they can actually insert microelectrodes to generate this membrane potential. So they applied this membrane potential, and in response to this membrane potential, the neuron shows a current. Now current means ion currents because due to the flow of current, ions, this current is generated. This axis represents time in millisecond, and this axis represents current in milliampere per centimeter square. Now one thing I want to mention, the current that goes in these directions from 0 to minus 1, we will call it inward current, and the current that goes from 0 to plus 1, we will call outward current. Now at first let me explain the first situations that had done by Alan Hodgkin and Andrew Huxley. That is, they actually fix the membrane potential of the neuron at minus 130 millivolt. Now, as because this membrane potential is less than minus 65 millivolt, that is the resting potential here, so we will call it hyperpolarization. So when they applied hyperpolarizations on a neuron, they find that the neuron generates a very brief current, a little current that is called capacitive current, and it only lasts for a fraction of seconds. So nothing very special occurred when they applied hyperpolarizations. When they applied depolarization membrane potential, that means they actually fixed the membrane potential of the neuron at 0 millivolt, that means higher from the minus 65 millivolt, then they observe the neuron response at first by generating a capacitive current that is outward in nature. It is also brief, but followed by a early transient inward current. Now why it is called early? Because this current is generated just after the capacitive current, so it is early. Now transient because it just remains or it lasts only for a few seconds, like 0 to 1 millisecond. So it is early transient inward current because the current goes in these directions. Now inward current means a cation come inside the neuron or an anion come outside of the neuron. So in any way, this is called inward current. Now after that, a delayed sustained outward current is observed. Okay, so applying depolarization membrane potential, they got this type of graph. This is very special because here you can see after the capacitive current, there is an inward current that is early transient inward current, and there is a delayed sustained outward current. Now why is it called sustained? Because as long as you keep this depolarization potential fixed, this current will going on. This will not decay. Okay, so this is delayed sustained outward current. Now outward current means cations can come out of this neuron or anion can come inside. Now from this first experiment, Alan Hodgkin and Andrew Huxley made a conclusion that membrane depolarization generates ion currents. Now I will explain the second experiment. Now let me explain the experiment 2, as because Alan Hodgkin and Andrew Huxley observed that neurons respond in depolarization potential to generate ion currents, so they applied the depolarization potential, membrane potential, but in a gradual manner. So they increases the membrane potential gradually. Like at first they applied minus 26 millivolt depolarization current, the depolarizations, and then 0 millivolt, then 26 plus 26 millivolt, then doubles it to plus 52 millivolt, and ultimately to plus 65 millivolt. Now they try to observe what happens to the ion currents when they increases the membrane potential gradually to the neurons. And they observe that the current not only flow, these two currents, the inward current and the outward current, not only flows, but it changes in magnitude. And this is the second experimental conclusions. I will explain it. At first, let me explain the early transient inward current. When they applied minus 26 millivolt on the neuron, they found that this much amount, see, this amount of inward current is flowing. But when they increases the membrane potential from minus 26 millivolt to 0 millivolt, they find that the inward current actually increases from this amount to this amount. So inward current increases 0 millivolt at 0 millivolt. But when they increases the membrane potential further from 0 millivolt to plus 26 millivolt, they observe that now the inward current is actually decreasing. So the inward current increases up to 0 millivolt. Remember this point. This inward current increases up to this 0 millivolt, but it starts to decrease when the membrane potential increases further. Like in plus 26 millivolt, it decreases. When they increases the membrane potential to plus 52 millivolt, they find that there is no inward current. It vanishes like this. And when they increases the membrane potential further plus 62 millivolt, 65 millivolt, the inward current becomes outward current. See here, it becomes outward in nature. So this is their observation. And in case of this delayed sustained outward currents, what they got? That this current behaves differently. Here, these currents actually when is minus 26 millivolt is actually flowing less than 2 milliampere per second square in this value. When increases to 0 millivolt, it touches the 2 milliampere per centimeter square current. When it increases further plus 26, it increases up to 4 milliampere per second centimeter square. When it increases to plus 52, it increases. So it basically the value of this delayed outward sustained current increases monotonically as we increases the membrane potential. That means these two currents is actually influenced by the membrane potential. Remember this. I will now explain. I will write the conclusion of the experiment 2 and then I will explain the experiment number 3. Now from the experiment 2, they got two conclusions. One is inward current increases up to 0 millivolt, then decreases. At plus 52 millivolt, this inward current stops, and at 65 millivolt, it becomes outward current. In case of outward current, it increases monotonically with increasing membrane potential. Now, by this experiment 2, Alan Hodgkin and Andrew Huxley do not know which ions is responsible for the generation of inward and outward current, but one hint they got. By Nernst equations, I have made a separate video on Nernst equation, how we can derive the equilibrium potential, so I'm not going into details, but just I want to mention here that Alan Hodgkin and Andrew Huxley got a hint as because they knew the ion concentration gradient of the squid giant axon, the inward concentration of sodium and outward concentration of sodium. They calculate the Nernst equations and they got the ENA, that means equilibrium potential for the sodium. Now what is ENA or equilibrium potential? That means in this potential value is approximate plus 55 millivolt. Mathematically you can got this value. So at plus 50 to 55 millivolt, the sodium ions will not show any net movement. That is the equilibrium potential, because ion concentration gradient helps in the movement of ions, but this potential will stop the movement of ions. And what exactly the Alan Hodgkin and Andrew Huxley got, that is at plus 52 millivolt, the inward current stops. That means they actually correlate these two values. So they try to understand that whether exactly this inward current is due to the sodium ion flow and the outward current is responsible for another ions. So they try to find out the answer and they perform the third experiment. I will explain it. Now let me explain the experiment number 3. By this experiment, Alan Hodgkin and Andrew Huxley want to confirm that sodium ion is responsible for the generation of inward current because they got a hint from Nernst equations. For this experiment, they kept the neurons at 0 millivolt membrane potential, that means a depolarization potential, because resting potential is minus 65, so increases up to 0 millivolt, and they find that the neuron is actually showing a normal current flow, that is inward current and outward current. Now they knew the concentration of sodium, external concentration of sodium in case of squid giant neuron, it is approximately 440 millimolar. So they kept the action at 460 millimolar sodium concentration, and this neuron showing the common current flow. Now when they kept the same neuron sodium free conditions, externally sodium free conditions, they observed now there is no inward current but there is an outward current. That means now sodium is actually coming out of the neuron because there is no sodium outside. So what will happen? There is inside some sodium, so sodium will come out. So from this experiment, and also when they re-establish the concentration of sodium at 460 millimolar externally, they find that the inward current comes back. But there is no problem with the outward current. See, neurons are kept at the sodium free conditions, the outward current is similar, and when they re-establish the sodium, only the inward current comes again. So from this experiment they actually confirm that for the generation of inward current, sodium ions movement is important, and sodium ions actually comes inside of the neuron by a channel that is actually controlled by voltage. So later it was confirmed that through the voltage gated channels, the sodium actually enters inside the neuron, and that generates the inward current. So now this experiments actually performed later in 1965 and 67. I want to mention here, and this experiment also confirmed that this two current is actually generated due to two different ions. As because this scientist also clamped voltage clamp at 0 millivolt, and then they actually applied tetrodotoxins in one situation and tetraethyl ammonium in second situations. See, in normal situations at 0 millivolt the neuron show this type of graph, but when they applied tetrodotoxin to the neuron, what they got, because tetrodotoxins block sodium channels, there is no inward current but there is common outward current is present. And when they apply tetraethyl ammonium that block potassium channel, they observe that inward current is present but there is no outward current. So from this experiment, further experiment, it was confirmed that inward current is generated due to the sodium movement and outward current is due to the potassium movement. Now outward means potassium will come out of the cells, and it is also influenced by voltage. So later it was confirmed that voltage gated potassium channels actually helps. So from these three experiments, what we got that this current is actually controlled by membrane potential or voltage. Then we got that this current actually changes in magnitude, and later we confirm that these two currents are generated due to the sodium and potassium, and ultimately we know that sodium through the voltage gated sodium channel, sodium comes in and that causes depolarizations, and after that potassium ions actually comes out of the neuron and that causes repolarizations. Hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching, and always keep learning.